Welcome family. Before us we have biology paper 2 for the year 2016 and this paper was written by the GCE candidate. So we have question 1 from section A. Figure 1.0 shows the setup to an experiment used to investigate the effect of a biological catalyst on the decomposition of hydrogen peroxide to produce oxygen. So we have our setup with potato disc inside and hydrogen peroxide, then the gas produced by bubbles. Then we have the table. 3 experiments were carried out under different conditions as shown in the table below. The first column consists of experiment numbers 1, 2, 3, volume of hydrogen peroxide, 5 cubic centimeter for each experiment. The number of potato discs used for experiment 1, 9, then 2 and 3, 5d6. The nature of potato disc used for this one, it's none in terms of boiled or raw. Then since we didn't have potato disc, that's why we just have this line here. Then we have boiled, then you have raw. Then in terms of volume of hydrogen peroxide, 1.5 cubic centimeters of hydrogen was produced for experiment 1 and 2. But 3 gave us 10.0 cubic centimeters. Then let's go to question A1. What is the general name given to a biological catalyst? So we are looking at the enzyme. Then 2, give a reason for boiling the potato discs in experiment 2. Now, what I want you to see the difference between the result for experiment 2 and 3, these two. So we can see that when the potato discs were raw, the volume produced was greater than the experiment 2, in which the potato discs were boiled. Why is the reason? So in experiment 2, the rate of reaction was slow. So was slow due to the absence of enzymes. There were no enzymes under experiment 2, and the reason is because the potato discs were boiled. Now, what was the effect of boiling the potato disc? Was to denature the enzymes. So say to denature the enzymes. So denaturing simply means to change the original shape of the enzyme so that the substrate can no longer fit. So that is what we are looking at here. Now we move on to question 3. Explain the difference in results obtained between experiment 1 and 3. So the result is in experiment 3, presence of enzymes, the reaction was fast, and because of that the volume of oxygen produced was greater than the volume of oxygen produced under experiment 1, where no potato discs were used. So say experiment 3 gave us greater volume of oxygen due to the presence of enzymes. So that is the case. So the reason, or maybe reasons behind using the potato disc for some of these experiments was that the board, we know that there should be enzymes inside. So we just want to compare what happens in a case where enzymes are used and in a case where enzymes are not used. So where the enzymes are used, we can tell from the results that the experiment gave us a greater volume of oxygen. Then we move on to question B. Predict what would be the result of Roman number 1 increasing the number of potato discs in 5 cubic centimeters of hydrogen peroxide. So the potato discs in our case are used as like the number of enzymes. So if we increase the concentration of enzymes, then the rate of reaction increases. Whenever the rate of reaction increases, then the volume of oxygen produced must increase. So we'll say increase in potato disc results in a greater production of oxygen. Then 2, heating the mixture of 5 cubic centimeters of hydrogen peroxide with 5 potato discs. Now heating here, we are just trying to increase the temperature. Now for this one, we are not going to increase the temperature beyond the optimum temperature for enzymes. No, we are just going to increase the temperature until we reach the optimum point where the enzymes can work effectively. So for this one, we are not expecting to have denaturing. No, we just want to give them the amount of energy needed for enzymes to work effectively. So this one will also result in a greater production of oxygen. This would result in increase of oxygen production. Then for question C, suggest 2 other characteristics of biological catalysis not demonstrated in A and B of this question. So for the characteristics that we've looked for this question, for this question, the first one is we've looked in terms of temperature, like heating. Then the second one is the concentration of the enzymes. So we can give now concentration of, we'll say, substrates. We've not looked into that. Then apart from that, we can also look at pH. We've not looked at pH. Even proteins, enzymes are proteins in nature. So we can now go to question 2. Figure 2.0 shows cells taken from a vesicular tissue in a plant. Okay. Then A1, identify the specialized cell in figure 2.0. So the whole cell, this is the phloem vessel. Now phloem vessel is just a vessel that transports manufactured food from the leaves to all parts of the body, which is the plant. Then Roman number 2, name one feature in figure 2.0 which enabled you to identify the specialized cells in A1 above. So what helped me to identify this as a phloem vessel is the presence of this E, which is the sieve plates. So say presence of sieve plates. We can also say presence of sieve pores. We can also say presence of companion cells. So all these are features or characteristics that we can use to identify a phloem vessel. Then Roman number 2, which letters on figure 2.0 corresponds to the following structures? The first one, nucleus. So nucleus, this is a shaded part, which is A. So we have A. Then cytoplasm must be a solution. So a solution or letter corresponding to a solution is letter D. So we'll have our D here. Then question B is we just want the function of cell labeled B. So what are the functions that we can give to a companion cells? So the first one, we know that the companion cell produces energy needed for, so companion cell produces energy required for transportation of food by the phloem vessel. So say the first one to provide energy. Energy for translocation. Then the second function that we can give to support, so we can also give to to support the sieve elements. Okay, we move on to question C. With reference to the specialized cell in figure 2.0, name the process by which substances are transported in the specialized cell. So this is translocation. So remember, translocation is the movement of manufactured food from the leaves to all parts of the plant. Then to suggest two substances transported by the specialized cell, so we'll have starch, then even proteins. Okay, we move on to question 3. Under question 3, figure 3.0 shows a diagram of a tooth. Then we have been shown a region where there is a tooth decay. We have part labeled F, we have sensory nerve, then gum here. Then A1, identify the type of tooth shown in figure 3.0 above and state its function. So this is based on the structure. You can see this area here, a V-shaped. This can be a molar or premolar. So let's just say premolar. Then the function is similar to a molar. So say for crushing and grinding of food. Then remember to label on the diagram the following parts of a tooth. The first one, blood capillaries. So these are found in the pulp cavity. So we can just see, label this side here. So we'll have blood capillaries. Then what else are we supposed to label? Dentine. So the dentine is just the region after the F, which is enamel. So this is our dentine here. Okay, we'll move on to question B1. Mention two elements or ions which enable part F to perform its function more efficiently. So part labeled F is enamel, so we want the ions that makes the enamel to be stronger so that it is able to break whatever that it should break. So the first one is calcium ion. The second one is phosphate ion. Then he suggests how tooth decay can be brought about. So we'll say by the remains of sugar from the meal. these sugars forms acids which dissolves the enamel. Thus we have tooth decay. Then in Roman number 3, give one way in which tooth decay can be prevented. So the first one, we can say by brushing regularly. Then by avoiding sugar containing meal. Okay, we move on to question 4. Figure 4.0 shows feeding relationship of organisms in an ecosystem. Then which of the named organisms represents the first one? A producer. The producers are always green plants. So in our case, we have grass. Then a primary consumer is any organism that feeds on the producer. So in our case, can be mice and grasshopper. Okay, then what is the primary source of energy in figure 4.0 above? So source of energy is always the sun. Then 2, C1, state the form in which energy flows from one trophic from one trophic level to another trophic level. So the form in which energy flows, this should be chemical energy. Then explain what happens to the energy named in C1 as it passes from one trophic level to the next. So say energy reduces as it passes from one trophic level to another. So one thing that you can master here is that only about 10% of energy is transferred to the next level, while the 90% of energy is lost via processes like respiration, locomotion, reproduction. Then D1, identify 2 organisms in figure 4.0 which are both secondary consumers and tertiary consumers. So both secondary and tertiary. So we have producer. Then here we have, this is a primary, so this can be a secondary. Then this is a tertiary. So let's go back. So 2 organisms, the first one is hawk. Then organism number 2, which can be both secondary and consumer. So apart from that, so the snake, so the snake also is our answer. Then using named organisms in figure 4.0, construct the longest food chain. So the longest food chain that we can construct from here is the one that we can have 1, so say 1, 2, 3, 4. So we have 4, or if you want we can say 1, 2, 3, 4. So the first one, we can look at grass. After grass, mice. After mice, snake. After snake, we have hawk. Or if we want, we can use grass, grasshopper, snake, hawk. So if we want, we can say grass, grasshopper, then snake, hawk. Okay, we move on to the last question, the fifth one. So table 5.0 below shows a cross between a brown male pig labeled pig B with two female pigs, pig A and white pig C. Then if you look at the first column, pig ABC phenotype, brown male, brown female, white female. Then the offspring, for B we have new for both white and brown. Then for offspring we have 4 white, 12 brown. So if this is converted in two ratios, we can say 4 divided by 4 giving us 1, then 12 divided by 4 giving us 3. So this is a 1:3 ratio. So 1 for white, 3 for brown. Then here we divide by 8 giving us 1. We divide by 8 also giving us 1. So this is a 1:1. Okay, so the reason why we are writing some ratios, these may be of help in terms of finding the genotypes. Then question A, explain why pig B had no offspring. So this is a male, yes, this is a male, and we know that male does not give birth. So say because pig B is a male and males do not give birth to offsprings. So it's only the female that have that ability. Now B, from the results in table 5.0, then using the symbols B for dominant allele and B for recessive allele, identify the recessive phenotype of the skin color. So the recessive is the one that is weaker in terms of expression, and it can only be expressed in a homozygous situation. So the recessive is white. You can see from the numbers that white is recessive, because even under the white female had some brown offspring. Suggest the genotypes of pigs A and C. So A, brown male, but this brown male produces white and brown mixture. So for male, there has to be capital letter B for brownie, small letter b for white. So this one has to be heterozygous. Then for genotype pig C, so for white to exist has to be homozygous, so we need to write small letters BB. So this is homozygous. And whenever we have 1 to 3 can only produce 1 to 3. Let's focus on 1 on 1. So we can have 1 to 1 whenever we have this type of genotype. So whenever you have heterozygous and homozygous for small letters, you may have this. So the key point is here. So we have 2, then 2. So 1 plus 1 is 2, also 2. So if you reduce, so this is, so for whenever you have 1 to 1, it means one genotype must be heterozygous, then the other one must be homozygous. Then using the genetic diagram, show the results of crossing pig B with pig C. So let's say parental phenotype. So say pig B, then pig C. So let's go to pig B. So pig B is brown female. So brown female. So under for that one, we are going to have also 1, 2, 3. How can we for this one to have 1, 2, 3, 1, 2, 3 brown female. So these two must be all carriers. So this one must be like this, even pig C with pig C also. No, pig C, what did we say pig C is? So pig C must be this. Yeah, sorry, I take this back. We are looking at the phenotype first. So pig B, brown. Then here we have white. Then we move on to parental genotype. Here we have said this. Then here it's this. Then parental gametes, we separate. Then random fertilization. Heterozygous for brown. Another heterozygous for brown. Then heterozygous. So say heterozygous, I mean homozygous. Okay, so let's see. Let me go back to, I need to check something. Brownie, so that one. This should all be capital letters. I'll explain why. So this all should be capital letters. See. So this will be PB, then PB, you have at BB. So the reason why this is in homozygous, because when pig B, what am I doing? Oh, why have I made this mistake? It's B and C. I made a mistake, another mistake. So I was correct at first. Sorry for this. So it should be a mixture. So B will have B will have B will have B. So B and C, this is our final answer. Now let me just go back to, okay, so we have our answers now as they were required. So that's it for this video. Thanks for watching.